Hello, I'm John Gribben, and welcome to day 2 at the completion of the first inaugural iwCAR-T cell meeting in very sunny Miami. I'm joined by my co-chair, David Maloney from the FMed Hutchinson Cancer Center in Seattle, and by Saar Gill from the University of Pennsylvania. An exciting day again, lots to talk about. So let's start with the session that you chaired this morning, and of course your own, as you described it, pet love, acute myeloid leukemia. Issues that you highlighted in your talk about the difficulties of targeting AML because of the effects on normal hematopoiesis. Do you want to talk about what you heard in the session today that kind of intrigued you and found interesting?
Yeah, absolutely. Thanks, John. So I think I started off the session with perhaps somewhat sort of a predictive and maybe theoretical considerations in terms of what might happen when we make good quality, potent CAR-T cells specific for myeloid antigens. With the underlying problem, I think that despite many years of looking, we haven't found a really great antigen that is specific to malignant myeloid cells and that is absent from normal cells. And I think that all speakers really came to the same point, which is there is no such antigen. We did see several different ways of dealing with that problem. And what I thought was particularly interesting in this session is that we are relatively early in the life cycle, shall we say, of CAR-T cells for AML, and so people have drawn disparate conclusions, I think it's fair to say, based on their experiences, and then I think that relates to how good the CAR-T cell product is that they've made, the patient populations treated. After all, we're still dealing with very few patients treated by each group. And so my underlying thesis, if you like, was that CAR-T cells, if they're going to be strong enough to eradicate AML, they're going to also be strong enough to eradicate the bone marrow, myelosuppression. And that was borne out by really exciting data, actually, from a Chinese group that used a novel design of a CAR, I think a very successful dual expressing CAR, where the T cells had a CD33 specific CAR as well as a CAR specific for another myeloid antigen that we have thought less about, but nonetheless has been described for the last few years, called CLL1. So each and every CAR-T cell that they made was specific for both of these antigens, and maybe that gave it different features than the single mono targeted CARs that everyone else uses. And they found a very high, impressively high response rate, by which I mean apparently eradication of the disease, but associated with very, very severe myeloablation, as one would sort of predict. In contrast to that, there are a few other groups that used either CD123 directed CARs or or NKG to do ligand-specific CARs that found less activity and less toxicity as well. And so I think at the end of
that same balance.
Same balance, yeah. Exactly. It's the same story, really, that if you want to go all out and eradicate AML, I think you should be prepared for the consequences. That's what I took away from the session.
Sure. So do you see, we'll come back and talk about the gene editing approach, because I want to talk about the myeloma first, and because that came up in both sessions, and we can think a little bit about that. So then we had a large number of talks on myeloma, and it's probably fair to say that's going to be the next disease in which we've got approved CARs. Efficacy, but relatively short durations of response, albeit in very heavily pretreated patients. What's your take on it?
My take on it was I think we heard about 5 different kind of clinical trials in some degree or the other, and all of them were associated with very high overall response rates in the 80 to 90% range, with some very stringent complete remissions, but everyone's kind of been out of shape a little bit about the durability, where it seems like on average people are relapsing in about a year on average, but some going as long as 3 years or a little more than 3 years. I think everybody was really taken by the anti-tumor activity, but puzzled by why when we get to a stringent molecular CR, why don't people stay in remission?
I was struck by the similarities to the pediatric ALL session yesterday, where you've almost got the same thing. Very high molecular responses, quite short duration of responses in some. Now, the difference there, of course, being that some of the patients are cured. And the question I asked for the ALL session yesterday is that to do with the fact that the leukemic stem cell doesn't express the target? And that's exactly the same thought I was having with BCMA for the myeloma session today.
Yeah, I mean, I think several people asked those kinds of questions, and I think we need to get better analysis of the relapses. Clearly, you can get BCMA negative tumors, just like CD19 negative escape. So I think that time will tell where this is going to fit. But it's clear that there's deep remissions in really refractory patients. And so the question will be, if you now back up to early stage disease or less disease, will they work better? Or is this a fundamental problem?
Are we going to have the same problem from a regulatory point of view that we've had in some of the other diseases, is that when it gets approved, it's going to be for this heavily pretreated population? I think the entry criteria were 3 previous treatments, even though the median on the study was 7. So you think we're going to expect the approval there to be what the protocol said for 3, or do you think they'll look at who was actually in the study and it will really push back where we can offer these CAR-Ts?
I guess it'll probably be 3, but with the requirement to fail IMiDs and proteasome inhibitors and daratumumab, at least in the United States, it seems like. The question I have as well is, if there already are trials that are starting that are going to look at, more of a consolidation approach where and we'll see whether those, if they work really well, then it's going to rapidly shift the population. But I think in the beginning, it's going to be these refractory patients. And the question is then how much do you value benefit for a year or 2 potential remission? I mean, some of the patients we've treated, I don't think would be alive another month if they didn't get treated, and they can go into really incredible remissions. So,
the other question that came up, and of course we're never going to get an answer for because we don't know what the cost is going to be, but the duration of response presumably in some way has to factor into what the cost can be if we start to think about cost per benefit ratio.
Yeah, there weren't too many takers on on providing an answer to that discussion, but I think that the valuation is really interesting in this population.
Now, the other thing I enjoyed about today was hearing a few presentations from China. Now, we've known a lot about the number of clinical trials and the sheer numbers of patients going on to the CAR-T cell studies in China. You already alluded to the kind of dual approach that they had in AML. And then we heard 2 different kind of approaches on almost the same study in the myeloma setting, an IST that is then, of course, the one that has led to the collaboration now between a Chinese-based company and a US company to try to, I like that slide of the bridge, bringing the 2 technologies together. Of course, what it also brought to mind was an issue I just want to touch on, because a lot of people usually find the regulatory stuff quite boring. But of course, one of the reasons the Chinese have been able to move so quickly is that they don't have to face the same sorts of regulatory hurdles. I'll come back and ask you about yours in the moment. But Erdmeyer talked about how long it took, and Dr. Patel from MD Anderson talked about 2 years of getting an IND through FDA approval, a regulatory approval. Is there something to be said for learning from the science that comes out of China, or are there ethical issues we should be asking about ourselves there?
I think that's a really good question. I mean, clearly things are changing in China. I know that they're increasingly requiring INDs for at least later phase studies than they were before. But you're right, you can do a clinical trial with just local IRB approval, and there's no IND requirement to go forward. And so I think clearly we saw some very rapid changing in clinical trials between patients, few number of patients, and that would be really hard to do in the United States.
I was looking at some of those timelines. I mean, once, for instance, your centers get a trial up and running, you can accrue patients fast, you can present it fast. But you saw some timelines today of the company set up the study, and a year later, the data is being presented at ASCO. That would be inconceivable for a US center to be able to deliver in that time frame, wouldn't it?
Yes, no doubt. I think the way it's done in China is very interesting. I think there are definitely pros and cons, and you're right, John, to point out potential ethical issues with that, since after all, while we do feel that the regulatory hoops we have to jump through in order to open a trial in the United States are onerous, at the end of the day, if the purpose is to protect the safety of the patients that we're treating, even on the first-in-human trial, then those are the hoops that we have to jump through. One would wish perhaps that they could be a little more efficiently done. But nonetheless, I think that is the difference. Can we learn from the way it's done in China? I mean, I think we can certainly learn from good quality, well-performed clinical trials with transparency, yeah.
The other way, of course, we can speed things up are the off-the-shelf CARs, the allogeneic CARs, and other types of approaches we heard about this afternoon. So what's your view on this?
Well, I think there's a lot of excitement and there's a lot of skepticism. I mean, and so it's clearly we've seen data now that with like the iwCAR-T drugs that those can be done. They can expand in some patients. They can provide some anti-tumor activity. The durability remains somewhat of a question. We think that those cells are ultimately going to be rejected. And in a case like a large cell lymphoma, do we need to have persistence, or is a flash in the pan, good enough? And so, they solve a lot of issues. It's a healthier T cell technically. It's coming. It can be made off the shelf, and so there's clearly some advantages. And you don't have to. If patients are too sick, they won't have to wait. And so there's a number of advantages. But I think those of us also see that even autologous T cells are rejected once they're gene modified and with a murine construct. And so even then the persistence isn't so good, and you can't retreat usually with those. So I'm pretty healthily skeptical still about this approach, because you do have to go to a lot of hoops to make a cell that's not going to cause GVHD, it's not going to get immediately rejected, and then you're dealing with a lot of immunosuppression and risk of infection.
Well, okay, so the last issue, of course, would be the regulatory issues. So you're now talking about CRISPR editing, TALEN editing. You're putting in 2 and 3 gene constructs. You then get a cell out, and we saw one slide saying there's 16 potential combinations of the cells that are there. What are the release criteria? What do the agencies look for? How much work is that got to do? And is that really the way that we've got to go forward?
I think the case of the 16 different populations is a rather extreme case, because there are several genetic modifications, and without going through the math, the more modifications, the sort of number of populations exponentially expands that you have to look for. So, as usual, release criteria are the degree to which you've established the transduction of interest, and then the degree to which you've knocked out the gene of interest, if that's what you're doing. With regard to specifically your question, I think, is what off-target editing have you achieved, or have you engendered? So there are important ways of doing that. It has to be an unbiased approach rather than an in silico. It has to be a screening approach. There are several ways of doing that. Guide-seq is one of those techniques. And then that has to be validated using deep sequencing, usually of those sites that are potentially thrown up by your screening. So, but then of course the obvious question is, if you do or do not pick out something in your validation, what does that mean if you do? And are you confident that nothing is there if you didn't? Because conceivably, it takes only one cell where you've introduced a missense mutation and important gene to cause mutagenicity. Having said all that, I know that we haven't, and I imagine other groups haven't seen an instance of that. And this is somewhat controversial, but I always think about what it is that we do, and we of course need to be extremely careful and comprehensive, but I always think about what it is that we do when we treat patients with chemotherapy, that we all very well know induces mutagenicity in their myeloid cells.
Well, look, we could spend the rest of the day talking here. We've run out of time, unfortunately. The good news is we've got so much to talk about. We'll have to have another meeting next year, which we all look forward to. So thank you very much for joining us, and thank you very much for your insights.